Hello, Lisa. Hello, Nitin. Hello. Hello. Lisa, can you hear me? Hello.
Hello. Yeah,
Yeah.
I was trying to.
Everything okay?
Yes, everything is
Okay.
okay. So,
Start around sharp 12 o'clock.
hello.
Hello. Let us start at sharp 12 o'clock.
Sure.
Recording in progress.
Hello.
Good morning, everyone. Welcome to the second session of the day, Super Resolution Microscopy and its application in quantitative biology by Dr. Nitin Mohan. I would now like to call upon Dr. Anu Philip, Head of the Department of Physics, Bharat Mata College, Srikakkara, who has been a great hold-up in our ventures for chairing the session. I welcome you, ma'am.
Good morning to all. Today, let us start our second section, of ICRAPS-2021. We, Bharat Mata College, welcome you all the participants to this section. Today, we are gifted with an eminent speaker, Dr. Mohan. Currently, he is working as an assistant professor at Department of Biological Science and Biotech Engineering, IIT Kanpur. Before joining IIT Kanpur, he has worked as postdoctoral research fellow in several universities around the world, which include University of Pennsylvania, United States, ICFO, Barcelona in Spain, ENS in France, and Academy Ciencia in Taiwan. His research interests include system design and development of super resolution microscope, single molecule imaging techniques for visualizing and understanding cellular physics in the context of proteins, intercellular transport and neurodegradation, cellular adaptation to nutrient sensing, etc. He is awarded his PhD in Nanoscience and Technology from National Academy of Taiwan, and he has completed his PG and UG from Mahatma Gandhi University, Kerala. He is also awarded Ramalingaswamy Re-Entry Fellowship, Department of Biotechnology, India, Academy Ciencia Postdoctoral Fellowship from Taiwan, Young Scientist Award from International Association of Advanced Materials. He has credited several international publications and books chapter in his credit. So he is such an eminent person, and it's my pleasure to welcome him to this conference. So on behalf of Bharat Mata College, I welcome you, Nitin sir. Today, he will deliver a talk on super resolution microscopy and its application in quantitative biology. Over to you, Mohan, sir.
Thank you very much, Anu, for that introduction. It was great. At the same time, it was a little bit overwhelming and weird because me and Anu used to be classmates, and you calling me with such official gestures is something something very unusual for me. So it was nice, and I'm very happy to be part of this venture. And so today, I think I can share my screen, right? I hope you can.
It's coming.
It's coming. And, I'm trying to. Yeah.
Yeah, everything okay.
Good. Great. So without any further delay, I would just jump into my talk. I hope I'm audible enough as well. So you could see my screen. So behind the title of my talk and everything, you can see a background where many things are moving, right? That's what you probably see. And you might be wondering what those are. So those are the cells that could represent the cells in your body. So these cells can be thought of as functional units of life. What does that mean? That means that some of the cells come together to form a certain organ or tissue, which will form your brain, while some other cells come together and form a liver or a kidney and make you the person who you are. So all these functional units have molecular mechanisms going on within them. So there are different molecules that come together to perform a certain function within the cell. And these molecules keep moving from one location of the cell to the other. So that basic movement is what we have captured using microscopy methods, advanced microscopy methods. And those images or videos that we get from these microscopes are the ones that I'm showing behind the screen that you see. So you could see that there are different organelles or different proteins that are moving within the cell. That's what you see there. So we will come to the details of it, why they move like this and what makes them move and what happens if they do not move according to certain physical laws. So that would eventually lead to different kind of diseases that you might get in your course of life, right? So that's why we are interested in probing the movement of these molecules from one location to the other. And to probe these are very tiny particles. They are very tiny molecules and proteins. To image them, we need high resolution or super resolution microscopy methods. So that's what we do in our lab. We develop these physical methods like microscopy methods, and we try and look into these cellular systems to understand how these molecules move from one location to the other. So before we jump into how these molecules move within the cell, I wanted to just give you some analogy for you to familiarize with what I'm trying to say, because I know most of these audiences do not belong to the biology background. So I'm trying to give you some analogy here. So think about the way we commute in our daily life. So we have highways which take us from our location of from your home to your location of function, that is your school, offices, colleges, or anywhere. So you go early morning, you get into a car or a bike or a bus or a truck, and then you go to your office space, right? And you do there your job, your function, and after that you come back home. So you commute bidirectionally every day almost to perform the function that you have. And this commute it has been enabled by different traffic rules that we have, right? So you have the traffic rules such as red, green, and yellow light telling us when to move, when to stop, and when to pause. Similarly, we have tracks for heavy vehicles versus lighter vehicles to make sure that there are no accidents happening in the roads, so that we can reach the right place at the right time without missing out on opportunities and function properly. Something similar happens in the cells. Each and every cell of your body right now, when I'm talking to you, is performing something similar. That what does that mean? I mean that the proteins and the molecules that are produced within the cell are moving from one location to the other. So think this is a cartoon depicting a single cell in your body. So there is something called nucleus in the cell where all the proteins are produced. The DNA that is required, the code that is required to produce the protein is present inside the nucleus. And the proteins are produced just outside the nucleus, which is almost towards the center of the cell. And these proteins and the organelles or the molecules have to move from that center of the cell to the periphery of the cell, probably because their function is somewhere in the periphery of the cell. You might have heard of organelles like mitochondria or lysosomes or endoplasmic reticulum. All these are names you might have heard in your basic elementary biology classes. So these organelles do not stay at one location. They move around the cell so that they can perform their function at different locations that they are required. Similarly, think about when you have food, the nutrients has to be taken up by the cells. So there are proteins and biomolecules that work together to process the nutrients that you consume. So those nutrients, those proteins might move from extracellularly, they come into the cell, and within the cell, they move from the periphery of the cell and they are taken towards the inside of the cell. So the trafficking is also bidirectional within the cells. The trafficking of lysosomes or endosomes or mitochondria or endoplasmic reticulum, these are different organelles within the cells. They all traffic bidirectionally. What I mean by bidirectionally is that they move towards the periphery of the cell as well as they move from the periphery towards the inside of the cell. And the analogy can be drawn like, how do they move, right? So they don't just diffuse around. Because if you think about it, these protein molecules are hardly 1 or 2 nanometers in size. And or hundreds of these mitochondria might be 100 nanometer in size. But the distance from the center of the cell to the periphery of the cell is in the order of tens of micrometers. That means they have to move from relative to their size. These organelles or proteins have to move hundreds or 10,000s of 10,000s more in terms of its length that it has to move. So a 10 nanometer molecule diffusing around to reach a 10 micrometer distance is something energy inefficient if you think about it. That's why these organelles are packed into vesicles, and they sit inside these vesicles, and then they are taken by dynein and kinesin motor proteins, and these motor proteins walk on these tracks called microtubules. I know all these terms might sound very new and very different for all of you, but the analogy is that these microtubules are tubule-like structures which can form tracks, and these tracks are analogous to the highways that we have. Now, these vesicles into which these proteins are packed can be thought about as you sitting inside a car. And the car itself can be thought about as these motor proteins. So imagine that you are the protein, you are the molecule that needs to get transported. You get packaged into these vesicles. And the vesicle, or the kind of vesicle that you got packaged into, is driven by now either a bicycle, so that becomes a different kind of motor. If it is a bike, a motorbike, that's a different kind of motor. Or if it's a car, that's a different kind of motor. And these motors just walk or run over these microtubule tracks. That's the analogy that I wanted to draw. To make things a little more easy to understand, I will go to this video. I hope I can play it. Yeah. So here you can see there is a vesicle into which these protein molecules are packed, and they are moving on a microtubule track like that. You can clearly see this is the motor protein that I'm talking about. The motor protein is literally by using the energy of ATP hydrolysis on these tracks. So if you want to see that one more time, so this motor protein is moving on the microtubule track. It is carrying on its head, it's carrying the protein or the vesicle that it is supposed to traffic. So here, this was the cartoon or the animation that I wanted to show. But in reality, if you do this under a microscope, here is one cell where I have labeled with some dye molecules. In green color, you are seeing the tracks. And in magenta color, you are seeing those vesicles that are getting transported within the cell. And if you look at under the fluorescence microscope, this is what you would see. You are seeing that the vesicles are moving on the magenta track back and forth. So this is the nucleus. This dark region is the nucleus where the proteins are produced. And this region towards the end is the periphery of the cells. So the molecules are moving back and forth as you see here. Now, coming back to the ideal scenario where everything is ordered and well placed, you get to move from your workplace to your home and back and forth, right? Because we have traffic rules that enable the smooth commute between your workplace and your office space. So now, but this is a dreamy situation. Most of the times we encounter a situation like that. Particularly if there are peak hours of traffic where you get a lot of congestion and traffic jams that happen. This is majorly because some of the rules are not followed, or for some people the rules do not exist. So it will always create congestion and traffic jams. And because of such scenarios, you might be late for the college, or you might miss out on an interview. So you are not being allowed to perform your function at the right place, at the right time, because of such congestions and traffic jams. Imagine such situations happening within the cell. So cell basically tries to commute its molecules back and forth, but there are traffic rules that exist probably within the cells that also make sure that such congestions doesn't happen within the cell when it is trafficking its molecules back and forth. But what happens if such traffic jams happen within the cell system as well? So these molecular traffic, are there molecular traffic rules that exist within the cell system? If such molecular traffic rules go wrong, do they lead to dysregulations or traffic jams? And does that have any impact on your health, such as your brain or your, when such traffic jams happen in your brain, do they lead to any brain disorders? So when such traffic jams happen in your liver or in your kidney, do they lead to metabolic disorders? These are some of the questions that we try to answer in our laboratory. So we are particularly interested in molecular transport within the brain or in the neurons. In your elementary classes, you might have seen this picture of a single neurons. Neurons mean if we just take your brain and isolate each and every cell in your brain, they would look like this. They are called neurons, and these neurons have specialized structures such as the dendrites and axons. And the axons are long, and then the end of the axon is depicted here. I will go back to my pointer. So yeah, the end of the axon is here. One of the end of the axon communicates with the dendrites of another neuron, right? So here I'm showing the dendrite of another neuron, and this is the terminal axon terminus of one of the neuron. So this is how the neurons in your brain communicate with one another. The signals are given from the axon terminus, and those signals are captured by the dendrites of the other neurons. So this is how the brain is interwired and the signals are communicated. But before these signals are communicated, the signals are coming from very much within the cell body or very much close to the nucleus. So the signals are basically biomolecules or biochemical molecules, right? And proteins. So they are all produced somewhere very close to the nucleus. They get packaged into the vesicles. They transport through these axons so that they reach towards the axonal terminus, and they can be released such that these molecules can be received by the next neuron, and that's how the communication is made. Now, think about, again, if you think about the size order, so these proteins or these vesicles are hardly a few hundreds of nanometers, and the axons can be meters long. Think the neuronal axon, the neuron that runs through your brain, connecting through your spinal cord, there is that neuron, the axonal length of that neuron can be 2 meters. So think about this, a protein or a biomolecule of 100 nanometer in size has to traffic around 2 meters, right? So there could be a lot of complications. There could be a lot of traffic jams. So nothing should go wrong here. If anything goes wrong, that can be detrimental to your brain. And how does that become detrimental to your brain? That's what we are trying to understand. So you might have all heard about Alzheimer's disease. I know that a decade before there was a Malayalam movie that came in where the lead hero is suffering from Alzheimer's disease. And the impact is that he starts losing his memory, right? So all of you are familiar with Alzheimer's disease. So here we are in the cartoon, it depicts on the left-hand side, you have a normal brain, and on the right-hand side, you have an Alzheimer's patient's brain, how it might look like. So the brain is really small compared to a normal brain. And why is that? Because the neurons started degenerating. You started losing the neurons. And that's why the brain becomes shrunken in size. So the postmortem reports of these brains of Alzheimer's patients also saw that there are protein aggregates. These are called amyloid plaques. Amyloids are proteins which can come together and form a big aggregate like that. So they saw these plaques or amyloid plaques within the brain, as well as neurofibrillary tangles that were also existing within the neurons in the Alzheimer's patient's brain. So why do these proteins tend to aggregate? Probably one of the reasons could be that these proteins were not trafficked to the right place at the right location, so that they get dumped at certain locations. And when that dumping happens much more frequently, they probably aggregate, and after becoming large aggregates, they couldn't be transported further. So this is one of the hypotheses that existed, that's why this is happening. And we wanted to test whether this happens in that way. The other reason is that we also see that, these tracks on which the trafficking happens, in an Alzheimer's patient's brain, these tracks starts to get disintegrated. These polymeric structures, which are called microtubules, the tubulin tracks, which should be otherwise very stable and sturdy, starts getting disintegrated in this Alzheimer's patient's neurons. So you remember these tracks depicts the highways on which the trafficking of the proteins happen. So if the track itself if the highway itself starts getting destructed, then you can imagine there is a lot of traffic jams or traffic blocks going to happen within the neuronal brain, right? Within the neurons in the brain. So this is something that we wanted to explore. Does it really happen that way? So what did we do? We took mice brains. So we cannot work on human brains. So we go to mice models. So we take mice brains. This is called hippocampus. Hippocampus is one of the parts of your brain. So we isolated the neurons from the hippocampus of a mice embryo, basically. And we treated these neurons with some nanoparticles, which are highly fluorescent in nature. These are fluorescent nanodiamonds, or some nanoparticles which are very fluorescent in nature. So these fluorescent nanodiamonds or fluorescent nanoparticles get integrated into the neurons. And then now you can see, with the virtue of this high fluorescence emission from the nanoparticles, we can track how these proteins are getting trafficked within the neurons. So we imaged these nanoparticles in the neuron for about an hour or more at a 2 minutes frequency. So in the right-hand side here, you can see in the video, I'm going to play the video. Yeah. So here you can see this, the gray region represents the axons and the dendrites, the intertwined axonal and dendrite structures within the brain of the mice. And these are the neurons, and these red dots represent the vesicles getting trafficked within the dendrites and the axons of the neurons. So you can clearly see that they are moving, they move and they stop for some time, and then they again move. So here you can see something moving. I hope you can see the video. So you can see some of them are moving, right? So if you analyze how these molecules move in a healthy mice, we can also compare this movement with mice which represent Alzheimer's patient's brain, right? Then we can understand whether the Alzheimer's people or the patients with Alzheimer's disease also have a problem with trafficking disorder. So how do we do that? So we know that there are certain proteins or certain mutations that happen to people who have Alzheimer's disease. So we create similar mutations in mice such that these mice will become representatives of these Alzheimer's patient's brain. And then we can compare that. So that's exactly what we did. So we created mice models which has protein expression levels compared to the levels of that of the Alzheimer's patient's brain. Then we conducted this kind of microscopy experiments to see the trafficking of vesicles and organelles and compared it with the healthy mice. That means mice without any Alzheimer's protein levels and mice with Alzheimer's protein levels, and then we saw that what happens to the trafficking. So when I say what happens to the trafficking, we are basically trying to see at what speed these or what velocity with which these movements happened, or whether they stopped for a longer time, or whether they were not able to move further. So these were the things that we tried to analyze. So how do we do this analysis? So here, for that, we use a single particle trajectory analysis for the microscopy data. So here you can see, this is the trafficking that I've told before, that these are the vesicles moving within the neurons back and forth, right? They are moving back and forth. So what we do, we reconstruct these tracks. That means that we have the position versus time, right? At each time frame, we have the position of this molecule. So those positions are put together as green dots like this for each frame. So the movie is like, say, several frames put together, right? So you get the position. So the position of the vesicles are put together in green dots. Each green dot represents the position of the molecule at that particular frame. And you get a track like that. This is the trajectory of the molecule. Now we are analyzing the molecule. So we want to analyze it. So this is how we analyzed it. So each of the position is given in green, and the distance between each of the position is marked as D1, D2, D3, etc. And the distance from the initial point to the final point is marked as capital D, or you can call this capital D as the displacement, and small d1, d2, d3 as distances between the points. Now, if you take a ratio of the displacement over the sum of the distances, that is d1 plus d2 plus d3, if the molecule was moving in a very straight line, then d over d1 plus d2 plus d3, or the ratio, is going to be 1, right? Because that value is going to be the same. Whereas if the molecule was moving in a very, not in a straight line, it was not able to move properly, that it started showing diffusive kind of behavior, then it will create angles between each of the distances, right? So D1 plus D2 plus D3 like that. So now you can think about it as, it's not being able to move very smooth as it would have moved in a straight line. It is making this kind of angles and moving here and there. In this situation, definitely the ratio of displacement over distance, sum of the distance, is going to be less than 1. So this ratio, we started calling it as the confinement ratio. And we put a condition that the confinement ratio should be over 0.8, should be close to 1. That's what we decided. And then we also put some angle criteria. That means if it doesn't make any angle between the distances, then it is good. That means the molecule is able to move in a straight way. But if it is keep making angles like this of over or 90 degrees, then we thought that maybe it is not being able to move in an easy format. And also the diffusion coefficient. So in this way, we are clear that the diffusion is not there. So whereas when it is moving like this, it's mostly a diffusive behavior, and it is not a directed motion. So we also think that it's not being able to move very well. So if the diffusion coefficient is over 1.45, then we also think that the molecule is not being able to move in a directed fashion or in a proper way. So putting all these conditions together, we analyzed each and every point in the trajectory. So that's what we did. So we ran such analysis over the points to see how we can get, how many of these points are easily moving and how many of these points that represent a congested motion or a kind of a traffic jam. So the green regions, after the analysis, we saw that the green regions represent a smooth movement, whereas these blue regions that come in between kind of represent a jammy situation where there's a pause behavior or a stop behavior happening. So that represents some sort of traffic jams. Now what we do, we get the biophysical readouts of these the run behavior and the pause behavior. The run behavior is calculated from these green regions, which will tell us the velocity with which the molecule was moving, and the run length. Run length means how much, for what length it moved between two consecutive poses, between two consecutive green spots, for how long it moved. That gives you the run length. Then the blue regions would give you the pausing behavior, means how much time it paused and how many pauses you can expect or you see within a single trajectory, that is your pausing frequency. So these kind of biophysical readouts kind of tell us what kind of movement we can get, right? What kind of moving pattern we can get. So we compare these biophysical readouts. So we'll get a trajectory from a mouse that is healthy and wild type. And we'll also get trajectories from mice which represent the Alzheimer's protein level expressions. So we can see what was the velocity or the run length of the trajectories from a healthy mice versus an Alzheimer's protein level expressing mice. That's exactly we did. So I think you remember the amyloid beta precursors that form the plaques in the neurons of the amyloid patient, Alzheimer's patients. So we know that we gave these amyloid beta molecules at 2 different concentrations. So here you can see a 0.5 micromolar concentration. Those represent the blue bars, and the magenta bars represent the higher concentration of the amyloid beta protein. So you can see that as the amyloid beta protein concentration increased, the velocity has decreased significantly, the run length has decreased significantly, and the processivity has decreased significantly, whereas the pausing behavior, such as pausing time, has increased significantly. So the black represents the control mice, or the mice that did not have any amyloid beta, and the blue represent the amyloid beta concentration at a lower level, and magenta represents the amyloid beta at a higher level. So you can see as the protein aggregation increases, the trafficking readouts started showing you the co-representative effects. Right. Similarly, we also looked for other proteins, not only amyloid beta. And from all these mice models, we constructed several mice models for several proteins that you could see expressed in the Alzheimer's patients. And we compared that for all of them, we could see that the trafficking readouts are altered in neurodegenerative disorders. So with this finding, we wanted to know why the trafficking readouts are altered, why the speed get reduced, why the pausing behavior increases. So that's what we wanted to know. So we came up with an idea that in these microtubules, these tracks that form the proteins that form the polymeric tracks called microtubules on which these motors are moving. These proteins are called alpha and beta proteins, alpha and beta tubulin proteins. And these alpha and beta tubulin proteins can get so many modifications like tyrosination, detyrosination, acetylation, etc. I'm not going into the details. I don't want to bore you with the biology of it. But what does it mean basically? It means that if you look into the cell, you can actually see different kind of tracks. So these different modifications will give you different kind of tracks. That means here you can see a certain track in magenta color, and you can see certain track in green color, because the proteins that represent normal tracks have been labeled with green, and the proteins that represent modified tracks have been labeled in magenta dye. So what happens, we kind of saw that there are clear, distinct set of tracks or highways that exist within the cell. So we started thinking whether these different tracks represent or can be correlated with the kind of tracks that we have for our commuting, right? We have tracks for heavy vehicles, we have tracks for pedestrians and bicycles. Why do we have this? Because we want to avoid accidents and collisions and smooth commuting of everybody. That's why we have different tracks. So we also started thinking whether cells also have different tracks for different purposes. Different motors can walk on different tracks so that they do not interfere with one another, and there's an efficient and smooth trafficking going on. And maybe that trafficking is disrupted, and that's why we see an altered trafficking in the Alzheimer's patient. That's where we were getting to. So first, we needed to understand whether these two different tracks will enable different kind of trafficking of motors in the cells. Basically, what I'm trying to understand is that when a vesicle moves on a green track versus a magenta track, whether it is going to be different. That's what we are trying to analyze with a microscopy method. But with normal microscopy method like this, I thought like I can zoom into this region and try to get more details of it to see how this trafficking happens. But the more I zoom in, I'm only getting blurry images. I'm not getting any more details, rather it is getting worse. So that's because it's a fundamental law of physics that light diffracts, and the diffraction has a specific absolute maxima size, and you cannot go below that size. And since the image is basically a diffraction pattern, you have an absolute size. So it's all because of diffraction limit that you get such blurry images, and you cannot zoom into something optically. So if you cannot zoom in optically, no matter how hard you try to zoom in digitally, it will only give you pixelated images. It will not give you a proper image. So this is a limitation that has been known for a century in the field of microscopy and still not been resolved until 2014, until 2010, where more and more techniques were developed to overcome this diffraction limit. And 2014, the techniques won a Nobel Prize in chemistry. So what are these techniques? And let's go into the details of that. So what happens, why this blurry structure appears like this is exactly what I said. So the protein that you, or the molecule, the dye molecule that you're trying to see is only like 3 to 5 nanometer in size, whereas the image of that dye molecule would look like 200 to 300 nanometer in size because of the diffraction limit. What does that mean? It means that, suppose this is your point source. Point source means the dye molecule that is trying to emit light. So this dye molecule is a point source. It can emit light radially in all the directions. And you are collecting you're trying to image this dye molecule with a microscope. A microscope is ultimately a lens, right? So you think the lens is like that, and when this light from the fluorescent molecule is captured by the lens will naturally form a diffraction pattern, right? You can see the diffraction pattern here, and the real image would look like that. And in the diffraction pattern, you have a central maxima, and the size of the central maxima is defined. It has a definite size, and that definite size is defined by the wavelength of this dye molecule and the numerical aperture of this lens. Okay, so imagine that you had that size is given by lambda over 2 times numerical aperture. Lambda means the wavelength of the light that you're using. So imagine that you used a very far red fluorescence molecule, which will emit, say, in the 600 nanometer range. So the wavelength is 600 nanometers. And normally the high-end microscopes these days have a lens with numerical aperture of 1.5. So the numerical aperture is 1.5 and the lambda is 600. So 600 over 2 times 1.5, that is 3. So 6 over 3 is coming to almost like 200 nanometers, right? So basically this size that you get is, if you are using visible light from the range of 400 nanometers to 700 nanometers, you are going to get a range of 300 to 400 nanometer for this central region. So a molecule that was only 3 to 4 nanometer, the image of it like this would look in the range of 300 to 400 nanometer. Okay, that's why we have, this is diffraction limit, and that's why the image looks as big as that compared to the real size of the molecule. Now, the even serious problem is that when, imagine if such 2 molecules are these 2 fluorophores, there are 2 such fluorophores and they are both sitting far apart. If they're sitting far apart compared to the diffraction spot, the diffraction limit of 300 to 400 nanometer, you can still see them as 2 different molecules, okay, 2 different fluorophores. But the moment they start coming close to one another, if they were sitting very close to one another, much below the diffraction limit of the molecules, then you stop seeing them as 2 different molecules. You will not be able to resolve them as 2 different molecules. You will start seeing them as a single blurry molecule. So that's a problem with diffraction limit. And that's where many of the techniques suffer, that you cannot get the images, you cannot image beyond the diffraction limit. So from this slide, you have to understand that the diffraction limit is 300 to 400 nanometers. And if multiple molecules are packed within 300 and 400 nanometers, you're not going to see them as separate molecules, right? That's a problem. So there are many imaging techniques. I'm just briefly discussing the scale at which you can image different imaging techniques and the scale at which you can image them. If you're imaging an organism itself, okay? It comes in the size of several centimeters to meters. So if you want to image our cells, then you can go for PET or ultrasound scanning and all those things, right? But if you're imaging the whole organ, again, you can use MRI or ultrasound scanning because they image in the range of centimeters to millimeters. Optical tomography can image you in the range of micrometers. But when we are imaging cells, you need micrometer range because cells are 10 micrometer to 100 micrometer range. So we can use microscopy methods like confocal microscopy, etc. But if you are using any microscopy methods to see what's happening within the cell, they are much smaller. For example, a virus entering the cell, you want to capture how the virus enters the cell. For example, coronavirus, or a bacteria that interacts with the cells, human body, or as we are discussing, the proteins that are moving within the cell, they all fall under the range of 10 nanometers to 100 nanometers. And they cannot be resolved because they fall below 500 nanometers. So 500 nanometer or above is what you resolve with normal microscopy methods. So to resolve two proteins sitting next to each other or two viruses sitting next to each other, you need to have microscopy methods called super resolution microscopy, which can break that diffraction limit. So there were several techniques that were developed in the period of 2008, 9, 10, 2008, 9, and 2010. During those years, several microscopy methods were developed that can go beyond this 100 nanometer range. And one of those methods were based on single molecule localization microscopy. So those imaging techniques were called STORM and PALM. I'm basically trying to focus on that in the interest of time. I'm not going to discuss about the other super resolution methods. So what is the idea behind STORM and PALM? STORM and PALM got the idea from single molecule localization methods. What do you mean by single molecule localization method? So if you remember this image from the previous slide, this was the image of a single dye molecule, right? So we know from this image, we know the 3 nanometer molecule, the image is like 300 nanometer. So we know that this 3 nanometer molecule is occupied somewhere here in this 300 nanometer image. This basically gives you a probability density where you can find the molecule, right? So if I want to accurately determine the position of the molecule from this probability density, what I have to, the image is basically the probability density, and I wanted to find what is the position of the molecule from the probability density. For that, we can just fit this intensity. This is fluorescence intensity. So we can fit the fluorescence intensity with the Gaussian curve. So the Gaussian would look like a mountain that is shown here. If I can precisely locate the peak of the mountain, that will give me the most accurate position of my molecule within this probability distribution, right? So that's the idea of the concept of single molecule localization. And how accurately I can measure the peak of the mountain depends on the signal to noise ratio. What does that mean? If my fluorescent molecule is very bright, it can give you a large amount of photons, then the peak of this mountain is going to be very, very sharp. That means if the peak of the mountain is very sharp, I only have probably one place to put the flag and say, this is the peak. Whereas if the molecule is not very bright, it is very dim, then the number of photons coming out is not very good compared to the background or the noise that is coming. So the peak of if you fit a Gaussian to that, you will find a flatter peak. That means if you climb a mountain and the mountain has so many, it's very flat, it's not very sharp, then you have so many places to put the flag and say that this is the peak, right? So you have more error with which you are locating the peak of the Gaussian. That's what I wanted to say. So based on this equation, that means that if all the signal parameter is given in the denominator, whereas all the noise parameter is given in the numerator. So this ratio tells us the error with which you are detecting the position of the molecule. So basically, you need very high signal to noise ratio to determine the position of the molecule from the image with high accuracy. So this is the concept of single molecule localization. And using this concept of single molecule localization, we can go and locate the position of all these diffraction limited image molecules that you see. All these are blurry images, right? Like all the dye molecules that are sitting in the sample are going to image at the same time, and then they all overlap with one another, and we are trying to locate the position of the molecule. But even with the concept of single molecule localization, it's very hard to find out where is the position of the molecule from these overlapping molecules, because they are all overlapping with one another. So the idea is to make only few of these molecules visible at any given time. If only this molecule is visible, this molecule, and this molecule, if these 5 or 6 molecules are visible, then I know that I am sure that they are not overlapping with anyone else. And then I can go and fit the Gaussian to all of these 5 molecules and then locate the position accurately. But if they all come together like this, then they are all overlapping with one another, and I will not be able to locate the position very clearly. So we needed to temporarily separate them. So that's exactly what we do in super resolution storm PAM imaging. So here is the microtubule structure that has been labeled with all these fluorescent molecules that I wanted to. For depicting it clearly, I have the yellow fluorescent molecules have been kept at distances, but you should imagine that they are densely packed and they are overlapping with one another. Okay. So all these fluorescent molecules will give me 300 nanometer images, and those 300 nanometer images will overlap with one another. And what I'm going to get a blobby, blurry image that is given in this red color. I know my molecule exists there, but I'm not resolving the structure of my molecule with such an imaging methodology. So what I do, I will allow all these molecules to blink such that, or now there are no molecules giving me fluorescence. Then I will randomly choose one of the molecules to give me the fluorescence. So it will give me an image. Then the fluorescent molecule depicted in yellow, when captured through the microscope, gives me this 300 nanometer image that you're seeing in the red color. Now from this image is now not overlapping with the image of any other fluorophore, right? So I can accurately determine the position of this molecule using the concept of single molecule localization. So I located the position of the molecule and I marked the location with a cross. Now I switch off that molecule. I randomly switch on another molecule. Then again, that molecule is not overlapping with any molecule. I can again locate its position using the concept of single molecule localization. And if I do this for all the fluorophores that I use to label my protein structure, then I can get the image of all the fluorophore like this. I can find the location of the molecule. I can put the location. And now if I put together the locations, I get the image of my protein structure, right? And this image now is no more limited by the diffraction. This is the concept or the idea behind super resolution imaging. And this is exactly how we do this. So here is the blurry image that you get from a normal microscope for the microtubules. And all the dye molecules that are used to label the microtubule is allowed to blink so that you only get a sparse subset of molecules at any given time. And these, whatever molecules appear, they have the position of those molecules are put together like this. And the positions can be put together or reconstructed to make reconstruct the image of the protein structure that was here. So that's why it's randomly you are making the molecules blink and then reconstructing the image from that. That's why the technique is called stochastic optical reconstruction microscopy, or STORM. Now, using this technique, this is the microtubule that we couldn't resolve before. With STORM, we can clearly resolve it. So that means if you look into the zoomed region, the normal microscopy, there are so many microtubules entangled with one another. Those entangled microtubules are clearly resolved with this technique. Now, coming back to our problem of two different tracks not being able to be visualized properly with conventional microscopy methods, we can clearly see the tracks differentiated from one another, the magenta track versus the green track, very clearly distinguished in this technique. Now we can measure when the molecule is moving in the cell, whether they were moving on a magenta track or a green track. We can clearly identify that And then we identified that and we analyzed the tracks as we discussed before. And we figured that the velocity was going down, run length was going down, and the pausing behavior increased when it was moving on a magenta track versus a green track. The magenta track was a detyrosenated microtubule. It's a modified version of the track. Okay, so we figured that this kind of a modification can make the molecule go slower. Basically, what we figured is that when they were moving, the movement is much more hindered on a modified microtubule. That's what we found out. And then we wanted to look into a mechanism for clearing the autophagy. That means that, like the same way we have, trash carrying, the recycling units that pass through the city, collecting the trash, and they recycle things for us so that the waste do not accumulate in the city. Similarly, cells also have a mechanism like that. So the cells will try and collect all the aggregates, all the unwanted proteins. They collect it and it is called autophagosome. The recycling unit is called autophagosome. See, autophagosome contains all the proteins to be degraded. They meet with the enzymes that are required for degradation, that is lysosome. So the autophagosomes and lysosome meet together and they degrade the proteins, and they can be used for forming new proteins. So this mechanism has to go on working properly in the cell. When this mechanism is not working, the unwanted protein starts getting accumulated, just like the waste getting accumulated in the city. And this again get transported using the motors and the microtubules. So we also figured that from using our technique of microscopy, we can see how these lysosomes and autophagosomes are interacting on these microtubules to degrade the proteins that are not required in the cells. So we also found that out. So you can here clearly see that the lysosomes are moving on these tracks, specified tracks that we just understood. They are moving and they are meeting with the lysosome. That's what we figured. So the proposed model of the study is that if you have two different kind of rods or tracks in the cell, they both traffic different kind of vesicles and motors, and there's a specialized kind of track that enables these degradation process within the cells. So we want to now put in perspective that all these proteins that are not getting degraded in the cell and causing Alzheimer's, there are two implications to it. That one, they are not getting trafficked properly. And we also figured that if they get trafficked on a specific kind of track, there is higher chance for them to get degraded. So we are now looking into therapeutic interventions whereby we can modulate this brain trafficking aspects such that we can increase the trafficking of these molecules on that specified microtubule so that these protein molecules can get degraded. So that's where we are going with our studies. And I would like to acknowledge the group that I was working before as a postdoc at University of Pennsylvania. This is at the group of Melchiel Academiali, my boss, where I did most of the studies that I showed you in this. And currently, I just relocated to IIT Kanpur a couple of years before. We are setting up our lab. And this is currently our lab. And we are continuing these studies in our laboratory. And I also wanted to advertise that, we are having a lot of research positions open. We are looking for motivated postdocs and PhD students and research engineers who can come into our lab and, contribute to the cutting edge science that we do. You can please refer to this lab website, www.bioresolutionlab.org, to find out more about what we do in our lab. And do contact me personally if you would like to join us for a research position. Yeah. With that, I would like to end my talk, and I'll be happy to take questions if there are
any. Now the time for question sections. Participants, if you have any queries, any questions, please unmute and ask. Otherwise, you can post in a chat box.
So I can see a question in the chat box. I think it is Marina asking me, can we use single particle trajectory to reconstruct chromatin organization? Wonderful. I think you are from a bio background, I hope. Definitely, that's another aspect that I did not talk today because of the time, definitely we can look into chromatin organization. We are currently looking into chromatin organization using super resolution techniques. Yeah.
Participants, if you have any questions, please.
Hello, sir.
Yes, Akshaya.
Sir, can I ask you what are the advantages of super resolution microscopy over confocal microscopy?
Yeah, so as we just discussed, in confocal microscope, you cannot go below 200 nanometer or 500 nanometers, right? So if two protein molecules that you're interested in looking in the cell are sitting close to one another, say they are only separated by 200 nanometers, you won't be able to see that. In confocal microscopy, you would see them together and you would infer a wrong conclusion that these two proteins are always together and doing some function. Okay, so unless you use super resolution microscopy, which can resolve, say, 20 nanometers, so you can see them separated. So in reality, those two proteins were separated, but confocal imaging will tell you that they are together. So that's a disadvantage of confocal versus a super resolution technique here.
Okay, sir, thank you.
Sir, one question. Sir, can we use super resolution microscopy in plant cells?
Yeah, why not? So anything that can be visualized using optical microscopy, that can be visualized using super resolution.
Sir, can I ask you one more question?
Here, please.
What do all super resolution microscopy technique offer the user?
Say that I couldn't hear you. What does?
What do all super resolution technique offer the user?
I'm not getting it clearly. Why? I can hear the last word user, but I'm not able to connect.
Sir, sir, sorry, sir.
Okay.
Share, repeat. No problem. Share.
So what do all super resolution technique offer the user? Benefits are, benefits.
Benefit is what I just told. So super resolution methods can resolve better. So it offers the user much more accurate, precise, and detailed information.
Okay.
Any other question?
Thank you, sir.
Good. I know you are muted if you're.
Yeah, I hope there is no more question. Yeah, Nitin's a wonderful section. Okay, I don't know how to express my vote of thanks in this section.
You don't
Anyway,
have.
anyway, you start with the normal transport section, and then next 15 minutes we go through the biology side. That side we are not familiar with terms, but we are always dealing that things. Okay, that I understand. Then we just go through the physics side. That's the optical microscopy, optical technique. Okay, that is the resolution limit. That is the diffraction limit. Okay, that things is our physics people daily using. Then you come to the 2014 Nobel Prize. That is in chemistry, the super resolution microscope. That is that indicate that this is an interdisciplinary field. I think the actual brain is physics people. Yeah. Without the microscopic technique, we can't show these things to the society. That is the main brain is physics people. Okay. Your talk is very informative and very interesting. That means you are all in all your slides, you show some technology. That is the movement, resolution, that is the microscopic picture we can experience from their slides. That's also very interesting and very helpful to our students to enter into that field also. We think that biotechnology is not for physics students, that's for biology students. That is different. Physics people can contribute more in biotechnology than a biology student. That I understood from this lecture. Within thank you very much. On behalf of Bharat College, I extend my sincere thanks for a wonderful section. Okay, thank you. And in other words, my classmates, my informal thanks. Thank you.
Sure. I would like to acknowledge the informal thanks more than the formal thanks.
Okay.
So thank you very much, Anu, for inviting me over here. And you rightly pointed it out that, I'm not, we both were physics students in the same class. So what we see currently in the field of biology is that people from other disciplines contribute much more relevant things to biology than the biologist itself. So that's why I wanted to encourage all the students who are listening, who listen to me today, if that inspired you even by a little, don't think that your field or your realm is closed to physics, but it is very open. So you should explore the field of biology. It's really fascinating. And anybody interested, one more time, anybody interested can look into my lab website. And if you're interested, contact me personally and we'll see what we can do together.
Thank you. Your lab name also Bio Resolution Lab. That's also depending on physics factor is there. Okay, thank you very much.
Thank you.
Okay,
Thank you very much. Bye-bye.
Okay, bye.
Thank you, Anu ma'am, for chairing the session. I express my gratitude towards Dr. Nitin Mohan sir for your electrifying words. Once again, I thank each one of you for your cooperation. Now, I would like to end the session with the great words of Dr. APJ Abdul Kalam. Teaching is a noble profession that shapes the character, caliber, and future of an individual. If people remember me as a good teacher, that will be a biggest honor to me. I request everyone to join the next session at 2 p.m. Thank you.
Recording stopped.